Hi, everyone. I'm Jennifer Nugent, the account manager for academic institutes on the West Coast, and welcome to today's CST Learning Lab live session. I'd like to introduce Dr. Rob McDonald, who is one of our flow scientists at Cell Signaling Technology. He received his PhD at Cornell University and has been with CST for 1.5 years. He validates antibodies and companion products for use in flow cytometry, supports marketing efforts through generation of flow data and associated content, and provides technical support for flow cytometry products. Today, we're going to discuss staining considerations when combining intracellular and extracellular readouts. During both live sessions, you will also have the opportunity to ask questions by typing them into the GoToWebinar questions area in your control panel. For those of you who are just joining us, welcome to the CST Learning Lab live session. Without further ado, I'll turn it over to our flow cytometry expert, Rob McDonald.
All right, thank you, Jen. Get things started here. All right, so welcome everyone. Today I'll be presenting the flow cytometry focused panel of Learning Labs Live. Today we'll be talking about some staining considerations when multiplexing with intracellular and extracellular readouts. So historically, flow cytometry has been known to be very amenable to multiplexing, but largely this has been limited to extracellular readouts. It's really common to use panels of CD markers on mixed cell populations and use that CD marker information to identify the different types of cells that may be present in your sample. You can also break it down by proportion of cells that fall into each subtype. Intracellular targets, on the other hand, have been largely studied by other techniques like Westerns or microscopy-based techniques. So why might one want to consider bringing in extracellular targets to a flow cytometry experiment? Well, intracellular targets regulate lots of major cell processes like apoptosis, cell cycle progression, and others. By including both intracellular and extracellular readouts in a flow experiment, it will allow you to get information on not only what types of cells are present in your sample, but also what each subtype is doing and how each individual subtype of cell might be responding to various stimuli. So when performing this type of experiment, your same general multiplexing considerations apply, just the same types of things you'd want to take into account when working with a panel of only extracellular antibodies. Namely, those things that will include prioritizing bright fluorophores. So you want to make sure to, whenever possible, use fluorophores that are going to give you the best separation between positive and negative populations. And to be able to get a sense of how bright different fluorophores are, you might want to consider searching for staining index values. And just to give an example of what it might look like to use a weaker versus a stronger fluorophore, here I'm showing some of our FLT3 products. A couple of them have been conjugated to weaker fluors, in this case Pacific Blue and Alexa Fluor 700, whereas one of them has been conjugated to a stronger fluorophore, Alexa Fluor 700. That's going to give you a better separation. So in blue is a negative cell line, and in green here is a positive cell line. The solid lines indicate cells that have been stained with the FLT3 clone, whereas the dashed lines indicate concentration matched isotype controls. So you can see that between the blue and the green lines for the Pacific Blue and the Alexa Fluor 700, we're getting maybe a little bit over a log of separation between the negative and positive populations. But with the Alexa 647 conjugate, we're getting a little over 2 logs. And it's important to note that these samples were all run on the same instrument, and the same concentration of that FLT3 antibody was used to stain our cells. The same types of cells were also used in this experiment. So this can be especially beneficial when you're dealing with cells that express targets more on a gradient say you've got low, medium, and high expressors as opposed to just a binary on-off. You're also going to want to try to minimize spectral overlap because this makes compensation easier. This becomes more important as you've got more fluorophores in your panel, and that'll happen as your number of readouts will increase. If you're only looking at maybe somewhere in the area of 4 to 6 targets at once, this is less of a concern, but if you're getting into the 8 to 10 plus range, this is something you'll want to pay more attention to. So I'll typically consult a spectrum viewer when I'm trying to get an idea of what the excitation and emission spectra look like for the fluors I'm considering in my experiment. There are a number of these around. This screenshot in particular is from fluorofinder.com. And so the excitation peaks for, in this case, 4 different fluorophores are shown in the colored dashed lines. And in the solid lines, we're looking at the emission peaks. And these are 4 different fluors that are excited by our 488 nanometer blue laser. And I've also included in black here the filter sets that are available on my cytometer. So looking at this first one in green, this is the emission peak for FTSE. In yellow, we've got PE. In the lighter red, we've got one of our tandems, PerCP Psi 5.5, and in the darker red is another tandem, PE Psi 7. So you can see that in this FTSE detector, we should be getting a pretty clean signal because we're only really seeing that emission peak from FTSE in this channel. Same thing really goes for this first tandem, the PerCP Psi 5.5. But if we're looking at our PE channel, we're going to have to compensate out signal that's spilling over from the PE channel, as well as from our second tandem dye, our PE Psi 7. You're also going to want to make sure to titrate antibodies. So at CST, we generally will give you a recommended concentration range for use, but this is not going to be ideal for every single panel out there or every different cell type out there. So I'd recommend titrating on a known negative and positive that should be ideally somewhat similar to the type of sample that you're looking at. And you're going to want to aim for the best signal to background, so the best separation between the negative and the positive samples that you're testing. Again, to make it easier to distinguish positive and negative signal. So once you've got that all set and you're considering starting your experiment, you're going to want to make sure to include appropriate controls. And I've listed just a few of the possibilities there. They can include unstained cells. You're going to want to include single stain compensation controls. These can be either cells or beads, as long as the signal is at least as bright as anything that you'll be using in your experiment. And then you'll also want to include fluorescence -1 or FMO controls, at least the first time you run your experiment. And this is going to help to give you an idea of where to set your gates. So one thing that is going to be a bit different about including intracellular readouts is that you'll have to fix and permeabilize your cells. This is because antibodies are too large to effectively cross the plasma membrane on their own, so they need a way to get in. Now, based on the reagents you use, it might affect some of the performance of your antibodies, and there are a few different factors here. I've highlighted some of them that seem to, at least in our hands, make a little bit more of a difference than others. But yeah, these include fixation reagents, fixation time, permeabilization reagents, the antibody concentration, and incubation and wash buffers. At CST, the vast majority of our products are going to be validated for use on cells that have been fixed with 4% formaldehyde for 15 minutes. We do have some variability in terms of the permeabilization reagents that we recommend, though. In most cases, we opt for 90% methanol, but there are some instances where we'll use other reagents like Triton X100. And antibody concentration, of course, this can vary quite a bit from clone to clone. For our incubation and wash buffers, we're largely using phosphate buffered saline with a little bit of BSA in there. I believe it's 0.5% as a blocking reagent. So I mentioned that perm reagents tend to have different strengths and weaknesses. So you've got a number of different options to choose from here. So of course, if you're looking at if you're working with live cells, that means they're not going to be permeabilized. You'll tend to get your best performance out of your extracellular antibodies using live cells, but you won't have access to intracellular proteins. So if you are interested in probing for intracellular readouts, you're going to have to fix and permeabilize your cells, and you can use selective or non-selective detergents as well as organic solvents as permeabilization reagents. Selective detergents like saponin and digitonin will remove cholesterols, and so this is good in that it doesn't disrupt other proteins on the plasma membrane to quite the extent as some of the other perm reagents, but you won't be able to access nuclear proteins because the nuclear membrane has a pretty low cholesterol content. Non-selective detergents like Triton X100 will simply dissolve lipids, and so this will give you a pretty good permeabilization, but you might lose some soluble membrane proteins in the process. And finally, organic solvents like methanol will also give you a very thorough permeabilization, but since they dehydrate your samples, they are going to be a little bit more disruptive to the epitopes. So even after fixation, there might be some conformational changes that happen, and that could impede binding of your antibody to its target. So your protocol options are going to be dictated by both your antibody and your target. In our experience, we found that most antibodies tend to be protocol flexible, meaning that they'll work under multiple protocol conditions, but there are some that are a little bit more finicky than others. So in this example, looking at phospho-S6 on cells that have been either untreated or treated with an inhibitor, they've been fixed with 4% formaldehyde and then permeabilized with either methanol, Triton X100, or saponin. And regardless of our permeabilization reagent, we're still able to see a good separation of our positive and negative populations. An example of something that's a little bit more finicky is this phosphostat1, where if we're comparing cells that are untreated versus cells that have been stimulated with interferon alpha to enhance that phosphorylation, we're really only getting a good separation of positive and negative populations on cells that have been fixed and then permeabilized using methanol as opposed to Triton X100 or saponin. So it's best to start out with a protocol from sources that you can trust. We'd recommend going with the protocol that's recommended by the manufacturer first, but you might also look for publications that have been well cited and well validated. You may also consider just asking around your lab or your department or field to see if anyone else has been having success using that same antibody that you might be interested in. If you're interested in using an alternative protocol, it's good practice to start out with one that you know works and then compare that alternate to that to make sure you're still getting a good separation of your positive and negative populations. So one of the big draws of being able to look at intracellular targets is being able to assess signaling events, and a lot of times these are due to post-translational modifications like phosphorylation events. So when you're doing this, you want to be careful about the time point that you're using to analyze your samples and how long you're treating your samples for. As many proteins are dephosphorylated or degraded soon after a signaling cascade is completed. Usually there's some type of feedback that happens telling it to either turn off or to essentially degrade. So depending on the pathway you're looking at, there might be some information you can glean from academic literature to give you a sense of how long you should be treating your samples for. But if that's not available, we'd recommend running a time course experiment. So just to give As an example of one site that's a little bit longer lived than another. Here we're showing PBMCs that have been stimulated with anti-CD3 and anti-CD28 for either 10, 30, or 10 minutes, 30 minutes, or 6 hours, and comparing them to untreated PBMCs as a control. So looking at phospho-76, we can see that it gets phosphorylated pretty quickly in response to that anti-CD3, anti-CD28 treatment. And there's still a good degree of phosphorylation even after 6 hours. On the other hand, phospho-S6 is a bit more short-lived. It still comes up really quickly within 10 minutes of treatment, but we can see that after 30 minutes, and especially after 6 hours, that phosphorylation event is essentially lost in most of your cells. So it's been turned off or dephosphorylated by this point. So one thing to consider is the order of your fixation and staining steps. One option for this is to simply take your live cells and incubate with your surface antibodies, then fix and permeabilize, and then stain with your intracellular antibodies. This is going to give you the best performance for your extracellular antibodies, but if you've got a short stimulation period that you're trying to work with, this might not be compatible with it because it's going to take you probably about an hour or so to go through that first round of staining. It'll also just take more time in general since you'll have to go through 2 steps of 2 incubation processes. You'll also need to be careful about the fluorophores that you're using on your extracellular antibodies, especially, well, really only if you're carrying through an organic solvent in the permeabilization step. This is because organic solvents will denature proteins like PE and APC. Small molecule fluorophores, on the other hand, like the alexafluors, will be able to survive that permeabilization step in methanol and without losing any of their fluorescent properties. Now, on the other hand, you could also consider fixing and permeabilizing your samples and then staining with both your extracellular and intracellular antibodies at the same time. This is going to save you some time because you'll only have to go through 1 staining step, but you have to make sure that your extracellular antibodies will work on samples that have been fixed and permeabilized. Some of them will only work on live cells. So to help illustrate that point, here we're looking at a couple of clones, CD3 and CD4, that still work well after the fixation and permeabilization process, whereas a couple others, this particular CD11C and CD14 clone, will no longer perform appropriately on fixed and permeabilized cells. So we can see that based on the live staining profile of CD3 and CD4, we're expecting 3 major populations, one that's double negative, one that's positive for CD3 and negative for CD4, and then a double positive population. And if we fix and permeabilize cells with saponin, methanol, or Triton X100 and then stain, we see that we're still able to get a decent separation of those 3 populations, and there's no additional major population that's jumped out. On the other hand, based on our live cell CD11C and CD14 staining, we expect 2 major populations, a double negative and double positive population, but we're starting to get some non-specific staining creeping up in cells that have been fixed with formaldehyde and then permeabilized with saponin or with methanol. If we permeabilize with Triton X100, we're losing almost all of our CD11C staining. And then we're also seeing that there's less intense staining of CD14. So here at CST, to try to help out researchers with that, we're doing some compatibility testing. So here we've got 4 of our different protocols: our live cell staining protocol, our fix methanol perm protocol, our fix Triton X100 perm protocol, and our FOXP3 fix and perm kit. So where we see a red dot that essentially means that this particular clone is not compatible with this protocol. A green dot means that it is compatible and it works really well. A gray means we haven't gotten around to testing it yet. And a yellow means that we saw some reduced performance, but it isn't quite at that level where we can say it just doesn't perform. You'll still be able to segregate out positive and negative signal, but these populations were likely going to be a bit closer together than if this dot was green. And we're also showing links to products that are available for each clone based on the fluorophore that they're conjugated to. This table can be found at cst-science.com/flow-table, and it's something that we hope to update a few times a year. So as a final point, I mentioned before that some fluorophores are impacted by organic solvents. So in this case, we've taken through some PBMCs, stained them with a CD3 clone that's conjugated to either a FTSE, which is a small molecule fluor, or conjugated to a protein-based fluor. And we can see that we're still getting good separation of positive and negative populations when these live cells are stained, or when the live cells have been stained and they've been carried through fixation and saponin permeabilization, or fixation and Triton X100 permeabilization. If we fix and carry them through a methanol perm step, on the other hand, the FTSE fluorescence is preserved, but the PE fluorescence is lost. This is because PE is a protein-based fluor that's getting denatured by that organic solvent. So you're going to want to make sure, if you do need to use protein fluors, to use them once all of the organic solvents have been washed out of your sample. So just to summarize things, when multiplexing with intracellular and extracellular readouts, your same general multiplexing considerations apply, and you've got a few additional decision points to make. These include when you want to stain your extracellular targets, and you can consult this compatibility table to get an idea of when you might consider doing that. If you can use your extracellular antibodies on fixed and permed cells, you might save some time by doing them in one step. But if your extracellular antibodies need to go on live, then you don't really have that option. You also want to consider which permeabilization reagent to use, and typically we just recommend going with whatever protocol the manufacturer suggests. This final point here really goes along with the first, deciding whether you'll need to use small molecule fluor versus whether it's okay to use a small molecule or a protein-based fluor on your extracellular targets. Just keep in mind that protein fluorophores can't be carried through an organic solvent. All right, so with that, thank you very much for your attention, and I'd like to take questions at this point. I think we're going to be prioritizing ones that are a little bit more general, but if you've got something that's more specific to your particular experiment or target, please feel free to contact us using our technical support forms. Thank you very much.
Awesome, thanks, Rob. All right, so we're moving on to the Q&A portion. Just a reminder, if you have any questions, please submit them through the GoToWebinar questions area. So we already have our first question. It's two-parter. How long should I stimulate cells for phosphoflow, and what cell line should I use as a positive control?
So this is really going to depend on what signaling pathway or specific target that you're looking at, because it's going to vary quite a bit. So I would recommend checking the literature on that particular pathway or target to see if there's been anything published. If not, you might want to consider doing a time course, and keep in mind that some targets are going to come up and achieve their maximum phosphorylation level really quickly within a matter of minutes, whereas others might take hours or even days to reach that maximal phosphorylation level. Yeah, in terms of what to use as a positive control, this again is going to depend on the signaling pathway that you're interested in. You might want to consider, I think if you type in Google cell signaling control treatments, you'll be able to come up with a partial list of various treatments and readouts to consider on our cell signaling web page.
Thanks. All right, next question is, how should I disassociate adherent cells for analysis by flow cytometry?
Yeah, so there are a few different options here, and basically they vary based on how harsh they are and how much time they will take to dissociate cells from your plate. One of the more harsh options is using trypsin. This will get your cells off pretty good, but you might lose some of your extracellular epitopes in the process because it's going to cleave some of those surface proteins. On the other hand, there's another reagent called Accutase, and that's marketed as a milder version of trypsin. We've had good success at preserving most of our extracellular epitopes when using Accutase to bring cells off plates. Another option would be using EDTA alone. This is probably going to be the gentlest option, but it will also take the longest to get the cells off the plates. And for really tightly adhering cell lines, it might not be a viable option. You could also potentially consider scraping cells. That's going to be another really harsh way of getting cells off. And once it's time to run your samples on the instrument, we'd recommend vortexing the tube for a few seconds to help break things up prior to running them through the instrument. And if your cells are particularly clumpy, you might consider running them through a cell strainer.
All right. Our next question is, do your antibodies work with fixed perm kits from BD?
So we don't actually validate our antibodies using kits from other vendors. So we can't say for sure that they would work, but for the most part, many of our antibodies do tend to be pretty flexible in terms of the protocols that they use, so it is likely that they would work. We just recommend that you do some testing on your own, comparing them to the protocol that we recommend before really going in and using them for larger experiments.
All right. Do I need to use an isotype control, and which one should I use?
So isotype controls can be useful when looking at cells that are expressing FC receptors. So those essentially will display some more non-specific binding than other types of cells because they'll interact with antibodies non-specifically. If you are interested in using an isotype control, you want to make sure to match the concentration of it to your experimental antibody, and you should also be using one that's conjugated to the same fluorophore. They should also match in terms of the host species, and ideally the heavy and light chain should match as well.
Great. Our next question is, how often should I repeat titration of my antibodies?
So I'd recommend titrating for every new antibody lot. So if you've got multiple tubes conjugated to, say, an Alexa Fluor 488, they're all from the same lot, you should be fine to just do that titration once and then feel free to use the other tubes from that same lot. The one exception to this would be with tandem dyes. Those tend to degrade a bit faster. They're much more sensitive than some of the other dyes. So you should repeat titration of tandem dyes for every individual tube of antibody, even if they're from the same lot.
Great. Can I use methanol instead of formaldehyde to fix cells?
Yeah, so it is possible to use methanol as both a fixative and a perm reagent, but when using methanol as the sort of dual purpose thing here, without using formaldehyde or anything else beforehand, you have a higher risk of losing soluble membrane proteins in the process as the lipids are being dissolved. And since you're dehydrating your sample before everything's kind of locked into place, your proteins may also have a bit more play in terms of being able to coagulate as they're dehydrated. So you might run into more of a risk of having epitopes covered up and inaccessible by your antibodies.
Mm-hmm. All right. Next question is CD marker staining can be affected by fixation and permeabilization, but can it be affected by fixation alone?
Yeah, so in our protocol compatibility table, we only include live cells versus cells that have gone through both a fix and perm step. But yes, it is important to note that fixation alone can impact performance. It can still have some effects on the epitopes of your surface antibodies. So if you're interested in running fixed and unpermeabilized cells, we'd recommend comparing the performance using a positive and negative line that have been fixed versus just that staining profile on live cells alone.
Great. How can we clearly differentiate live cell, apoptotic cell, and necrotic cell from a single cell suspension using flow cytometry?
So for live cells, you could consider using annexin 5 and PI for this purpose. Live cells should be negative for both of them, whereas early apoptotic cells should be positive for annexin 5, as this will bind to phosphatidylserine, which is normally only on the inside of the plasma membrane, but in the early stages of apoptosis, it translocates to be present on the outside as well. Early apoptotic cells will also still have their membrane intact, so they shouldn't be positive for PI, whereas necrotic cells will be positive for both PI and annexin 5. If you want to you do this type of analysis on cells that you're fixing and permeabilizing, you could consider replacing that PI with a ghost dye, and this would be added before fixation. And then after the cells have been fixed and permeabilized, you could probe with an antibody against an intracellular apoptosis marker like a cleaved caspase-3.
Great. All right, so it looks like we have time for just one more question. This is asking, do you need to use different fixed perm protocols for different cell types, such as epithelial cells versus lymphocytes?
We haven't seen this impact performance. Usually we recommend a particular fix and perm protocol based on the antibody rather than the cell type. We tend to perform validations on a wide range of cell types, both suspension and adherent lines, and we haven't noticed the cell type having a big impact on antibody performance.
Great. Well, thank you, Rob, and thank you everyone for joining us today for the Learning Lab Live session. If we didn't get a chance to get to your question, we'll reach out to you via email following this. If you have any additional questions after this webinar, feel free to reach out to our tech support or your local account manager, and they'll help connect you with tech support. Stay safe and well, and have a good day.
Thank you very much, everyone.